Hello, students. My name is Jatin Dewan, and today we are going to understand biotechnology, principles, and processes. So, biotechnology deals with techniques of using live organisms or enzymes from organization to produce products and processes useful to humans. According to European Federation of Biotechnology, the integration of natural science and organism, cells, parts, and thereof molecular analogues for products and services. It has applications in agriculture, food processing industries, medicine, diagnostics, bioremediation, waste treatment, and energy production. So, principles. What do the principles tell us about genetic engineering and aseptic techniques? Genetic engineering may be defined as the techniques to alter chemistry of genetic material into the host genome and altering its phenotype. Techniques of genetic engineering include creation of recombination DNA, gene transfer into host organism, gene cloning. Aseptic techniques would include it is the maintenance of sterile ambience in chemical engineering processes to enable growth of only the desirable microbe in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. The tools of recombinant DNA technology include restriction enzymes, and then thereafter they are of two types, endonuclease and exonuclease. So in the year 1963, two enzymes were discovered from Escherichia coli, which is also known as E. coli, which restrict the growth of bacteriophage in it. One of these is added methyl groups to DNA and other cut the phage DNA, which is also known as restriction endonuclease. So now let's have a look at the two kinds of nucleases. One is exonuclease and the other one is endonuclease. So exonuclease cut the DNA at the ends and endonuclease make cuts at specific points. So endonucleases always cut at specific location having a specific sequence of base pair called as recognition sequence or palindromic sequence. Example, E. coli or E. coli recognizes only the following sequence GAA-TTC CTT-AAG, which is fifth to third and third to fifth end. They create sticky or blunt ends. Have a look here. They help in generating DNA fragments with precise ends. These precisely generated ends can be pasted to similar ends in vector molecule that is self-replicating. The process of pasting is called ligation and require DNA ligase. The product after ligation is called DNA, recombinant DNA molecule or a clone. Then the second part of this is the cloning vectors. So the plasmid and bacteriophages have the ability to replicate within the bacterial cells independent of the control of chromosomal DNA. Alien DNA linked with the vector multiply its number equal to the copy number of the plasmid or bacteriophage. There are several features responsible for it: origin of replication, selectable markers, and cloning site. Plasmids and bacteriophages are used as vectors. Plasmid is an independent circular and self-replicating DNA molecule that carries only a few given genes. Look at this, the plasmid map. Now vectors for cloning genes in plants and animals. So let's have a look of vector for cloning genes in plants first. Agrobacterium tumenificens can cause the plant disease called as crown gall by transferring specific genes to dicots. It contains a large plasmid called Ti plasmid, which can deliver tDNA to transform normal cells into a tumor and direct these tumor cells to produce the desired chemicals. Have a look here. This is the Ti plasmid and this is the tDNA region. So these are cytokinins present here, and then similarly the other things are present. Vectors for cloning genes in animals. Retrovirus in animals has the ability to transform the normal cell into cancerous cell. Retrovirus are first needs to be disarmed and then used to deliver genes into animal cells. The next topic is competent host for transformation with rDNA. DNA is a hydrophilic molecule. As we know, it attracts towards the water. It cannot pass through cell membranes. So in order to force bacteria to take up the plasmid, the bacterial cell must first be competent to take up the DNA. Then the bacterial cell is treated with divalent cations such as calcium, which increases the efficiency of DNA uptake by the bacteria. Recombinant DNA and the bacterial cells are incubated in ice, followed by placing them briefly in 42 degrees centigrade. Heat shock, other word for 42 degrees centigrade, and then putting them back in ice. By microinjection, the recombinant DNA directly is injected into the nucleus of the animal cell. The plant cells are bombarded with high velocity, but with micro particles being present of goldenate or tungsten coated with DNA in a method known as biolistics or gene gun. The disarmed pathogen vectors, which when allowed infecting the cell, transfer the recombinant DNA into the host. The process of recombinant technology includes isolation, fragmentation of DNA by restriction endonuclease, isolation of desired DNA fragment by gel electrophoresis, ligation of DNA fragment with the vector by DNA ligase, transferring the recombinant DNA into the host, culturing the host cells in a medium at large scale in bioreactor, and then further extraction of desired product by downstream processing. So let's have a look of first one, that is isolation of genetic material. So as we know, DNA is a genetic material, and then bacterial cell wall is digested by lysosome. Then after the digestion, plant cell wall is digested by cellulose or pectinase, depending upon what cell wall is present. Either it is bacterial cell wall or plant cell wall. The fungal cell wall is digested by chitinase or chitinase. RNA of the cellular content is digested by ribonuclease. Proteins are removed by proteases, and purified DNA ultimately precipitated out after addition of chilled ethanol. The precipitated DNA is separated and removed by spooling. Then amplification of gene of interest using PCR, that is polymerase chain reaction. It multiplies the copy of gene of interest, which can be synthesized in vitro. PCR includes the following steps. So PCR includes a couple of steps. First one is denaturation or denaturing stage, which happens at 94 degrees centigrade to 95 degrees centigrade. Double-stranded DNA is made with single-stranded, is made to be single-stranded, and each single-stranded DNA is called template strand. Then the second stage is annealing stage, which occurs around 50 degrees to 56 degrees centigrade. Two sets of primer, that is the small oligonucleotide chain that are complementary to the DNA at third end of the DNA template, is added to the medium. Then the final step of amplification includes extending stage. Extending stage is deoxyribonucleotide triphosphates are added in the medium. Taq polymerase catalyzes the polymerization reaction using nucleotides extending from the primer towards fifth end of the template. Taq polymerase is a thermostable polymerase isolated from a bacterium called Thermus aquaticus. It catalyzes polymerization reaction at 74 degrees Celsius or 72 degrees Celsius. Then insertion of recombinant DNA into the host cell. So insertion happens after the obtaining. The protein encoding gene expression is obtained in a heterogeneous host, which is called as recombinant protein. The host is cultured into a continuous culture system provided in bioreactor. A bioreactor provides optimum growth conditions such as temperature, the pH value, substrate, salts, vitamins, oxygen, and etc. required for it. Bioreactor converts the raw material into specific product and specific enzyme. Then obtaining the foreign gene product, as I've explained earlier. So after this, the downstream processing happens. After biosynthesis inside the bioreactor, the product has to be subjected through a series of processes before it is ready for marketing. The process includes separation and purification, which are collectively referred as downstream processing. The product has to be formulated in suitable preservatives, such as formulation has to undergo through critical or clinical trials, as in the case of drugs. Thank you so much, students. I hope your doubts have been cleared by now. And if you have any further doubts or any questions, please don't hesitate yourself to drop in the comment below. Thanks.